Just a second. Just. One second. I think there is some technical problem with the. Yeah. Can you see my screen now?
Yes, sir.
Okay, right. So, Sir Charles Scott Sherrington, the person who was behind the reflex theory, he started his works in 1860s, and he had got his Nobel Prize in the early 1900s. And his works has been very monumental, and it has given us very wonderful understanding about how the nervous system might be functioning. So he developed a set of ideas based on his experimental findings, which he collated as the reflex theory. This reflex theory has been used extensively by physiotherapists of the past 100 years. We had many treatment techniques coming out of this reflex theory. The famous ones are the Roots approach, the Bobath approach, the Brunstrom's approach, and the PNF techniques. So he had summarized his findings in the so-called Silliman's lecture, and in his monumental work, The Integrative Action of the Nervous System. So with this brief introduction, we saw that the basic idea behind this is he tells us the nervous system functions by responding to stimulus, and the integration of the function happens by coordination of various reflexes. So if you look at the nervous system, he tells that the nervous system functions by coordinating multiple reflexes, and stimulus forms the basis for all the movements and the responses we are producing currently. So this is the famous reflex model, and till date when we teach, this forms the basis of many of our early periods of teaching for the students. Now, what he tells us, the simple reflex, as we see in a knee jerk, whenever we hit the patellar tendon with the knee hammer, the response we see is a very simplistic view of how the what is a reflex and how the nervous system functions. But rather, he tells that the beauty of production of the movement is in compounding of the reflexes. However, in order to understand the compounding of the reflexes, he tells we need to first understand the features of a simple reflex. And whenever we tell about a movement, he says that it is never a single reflex acts, it's always multiple reflexes are acting with each other. Some of the common examples which he has taken in his studies of animals, including cats and dogs, where for the simple reflex, for understanding of the simple reflex, are flexion reflex, the cross extension reflex, and the scratch reflexes. Now, we all know that flexion reflex is whenever you have a noxious stimulus in the, whenever we have a, uh, whenever we have a pinprick or thorn is pricking under the feet, we immediately withdraw our leg, and that is called as a flexion reflex. When we withdraw our leg, the opposite leg also extends. It's called as a crossed extension reflex. If both the limbs are going into a flexion reflex, we will eventually fall on to the source of problem. That's the pin or a thorn, right? Then if a noxious stimulus, if it occurs in some part of the body, which is very different from the feet, then what happens is the animal tries to remove it from its place of origin by by doing a repeated scratching of that particular place in the body. And this has been classically told as a scratch reflex. So scientists from then on, even slightly before and till now, they study these reflexes in order to understand important functioning of the nervous system. They take it as indirect clues as to the functioning of the nervous system. We also see that in many of the clinical conditions, like cerebral palsy or any other developmental disorder, we consistently see that we evaluate the reflex and we see whether the reflex is normal or it is abnormal, and how it has integrated with our own body movements in order to interpret whether the amount of damage to the nervous system and where exactly the damage could be. That's how we have been looking into it. So study of reflexes has always been an interesting part, and Sherrington predominantly has done majority of his experiments on simple reflexes using these three reflexes. Right? So now coming to compounding, he says that the compounding can happen by two ways. One is simultaneous coordination, that is one reflex and another reflex can act together. That's called a simultaneous coordination. But he tells that whenever they come together, the function of one reflex does not oppose the functioning of the other reflex. And he tells that this is the most crucial beauty of the nervous system. And this is what he calls as integration of the reflexes. Also, sequential coordination is very well known. We always know that there have been some writing reflexes and equilibrium reflexes which come one after the other stage during the development of the child from its younger ages to it reaches the age where it can move around very freely using adult-like movement patterns. So that's called as a sequential coordination, and that also flows on very smoothly. So he tells that the compounding of the reflexes helps us in bringing out different types of movements. Okay, am I clear and up until this point of time?
Sir, if it is okay, can you just once more explain the sequential coordination?
Yeah, sequential coordination is when one usually what we see is a reflex comes at one point of time, and then the reflex, a second reflex can take over. Okay, right? To give an example, um, mm, we have seen previously successive induction and reciprocal inhibition, right? So, uh, so what happens is one reflex acts, and it slowly it goes off, and then the second reflex occupies its place and it produces its action. That's what we mean by sequential coordination. So again, he tells that it's a integration wherein the coordination is very smooth. There is no contradictory functioning between the two reflexes. Am I clear
Okay,
with
okay.
this?
Yeah.
Right. So that's
Thanks.
the simultaneous and sequential coordination happens. Okay, right? So now he had talked about the simple reflexes, and he tells that simple reflex is just example. It doesn't occur or it doesn't help in any way. But understanding a simple reflex is very much essential for us to understand about the compound reflexes. So for that sake, he has spent 3 of Silliman's lecture in explaining and in detail about the simple reflexes. Okay, so now we'll move on to the simple reflex, lecture 1. All right. So in the first part of his talk, he compares a simple reflex with conduction in the neuron. Okay, what till then people were knowing is how when we are giving a stimulus to a neuron, how does it produce a response? That's what people have been knowing. And then he has compared both of them, and he found that, to his surprise, that many of the definitions and many of the ways in which people said that the neurons function was not what he saw when he had stimulated at slightly different point in the nervous system, and when he took out the responses from the other end of the nervous system. And whenever he studied, all right, he what he did was he had hmm, yeah, one second, just yeah. So what he has found is he used a set of experiments. Mainly in these experiments, he had given a stimulation to the nervous system and he studied the output. And the stimulation what he gave were of different kinds. He gave mechanical stimulus, he gave electrical stimulus, and he studied how the animal was responding. And what he found was when he had studied these things in a person who is having a complete brain, that is an animal which is having a intact brain, and when he had cut the brain across different regions, like between the spinal cord and the brain stem, or between the brain stem um, and the base of the cerebrum, or when he had cut out only the cortex and left the other parts to remain intact, he saw that the similar stimulation started giving a variety of responses. So there were differences when the spinal level there was a cut. There were differences when brainstem level there was a cut, and at other levels. At the same time, he also saw that many of these responses which were coming out also had some similar properties. Okay, right? But eventually, all these properties, whatever he had seen in these reflexes, were very different from that of a simple nerve trunk stimulation. Okay. All right. So, what were the differences he found? So, Sergio had started a presentation and she had given a lot of features. All right. So, we will look into it again. The first difference between nerve conduction and reflex conduction is in a nerve conduction, whenever you give a stimulus, the response comes very fast. So, that's the faster speed of conduction. Rather than a reflex, the speed has been slower. This is what he called as there is a time gap between the stimulus and the response, and it was called as latency. Right. The next one is there was a close correspondence between the stopping of the stimulus and stopping of the response. Okay. What it means is um, when you stimulate a nerve and when you stop the stimulus, you immediately see that once the stimulus is stopped, the response also stops. That's what he meant by close correspondence between stopping of the stimulus and stopping of the response. However, when he analyzed the reflex arc, he found that there was less correspondence between time of stopping of stimulus and stopping of response. This is what he found was when he had stopped, he saw that the response was continuing for a certain period of time, and he called this as after discharge. The next one is he saw that whenever a nerve was stimulated, all the stimulus produced a response. But in a reflex conduction, there was a particular rhythm, which means that all the stimulus was not producing all the responses. So he said that there is a rhythm to the response which comes out of a reflex arc, and it can be variable. The fourth one, the rhythm of stimulus doesn't come produce a similar rhythm of response. He coined a term called refractory period, and we saw in the last class a very detailed explanation of what's the refractory period, right? The next one is he found that whenever he stimulated the nerve, right, once a particular intensity is reached, after that, every stimulus produced a response. So the nerve conduction followed a all or none phenomenon. But in a reflex conduction, whenever he started stimulating it with a very minimal intensity, and then he started increasing the amount, the intensity of the stimulus, he saw that the magnitude of response was varying all the time, right, from very less to very high. So this was in contrast to what he found in a nerve conduction. The next is he found that there was an easy passage of current through the nerve. The nerve was not giving much of resistance, but when the same impulse was passing through the reflex pathways, he saw that there was much resistance to the conduction. But when he increased the intensity of the stimulus, he could see that it is pushing forward, and he was able to get the resistance to reduce, and it was conducting faster and faster. That's the other difference he saw, right? The other one he saw was conduction can pass in both the directions, that is in the direction of normal physiological conduction, which we call as orthodromic, and also in the opposite direction. But in a reflex arc, he saw that the stimulus can pass in one direction only, and it was not reversible, right? And finally found that there was no fatigue in the nerve conduction. That is, when you kept on giving the stimulus, it kept on giving the responses, but in terms of reflex conduction, it was fatiguing. Okay. Beyond this, he also saw that there were few things which were related to the previous points, whatever we have said. Nerve conduction had a standard threshold value of minus 90 millivolts. It's not microvolts, it's millivolts. Okay, right? But reflex conduction had variable threshold values, which meant that some of the reflexes could be elicited at very easily. Some of the reflexes required a larger amount of current in order to elicit it. So that is other feature he saw as different. And finally, there was no refractory period in the nerve conduction. Okay, right? But when it came to the reflex pathway conduction, there was presence of higher refractory periods as compared to that in nerve. When I say no refractory period, minimal refractory period, but it was very much higher in case of reflex conduction. All right. So this was related to the previous point. So these were the major differences he saw, and so he understood that the when we are stimulating a single neuron, right, and when we are stimulating the nervous system, they both are not similar; they are different. Which meant that the collection of neurons, when you collect them, when they all start functioning together, they are not just merely adding up together, but they are interacting together, and they are coming out with a very different type of functioning and varieties of functioning. So he understood that it's not just a simple addition, but it is a compounding of the functioning. Okay. And finally, his major issue was to understand where does many of these properties arise from. Say, for example, the latency, the after discharge, the place of after discharge, the place for refractory period, because these were not seen in a nerve trunk. So he said that within the nervous system, when he had analyzed the anatomical structure, he found that there were predominantly four common things. One is cell body was there. Second one was its projections, which are the dendrites. Then the third one he found was a unique type of cells which were not participating when we are giving a stimulus. It's called as neuroglia. And he also found that between one nerve and the other nerve, it was not continuous, but there was a surface of separation. Okay, right? So he was not sure as to which of these is, are the reason for the differences in the functioning. And he deduced that by looking at the neuron, that cell body and dendrites are common between the peripheral nerves as well as the central nerves. So that may not be the reason. And then he was thinking, could neuroglia be the reason, or the surface of separation could be a reason? Eventually, he came to a conclusion that neuroglia is not going to perform a major function. It might be having something else. And so the surface of separation could be a reason for the differences in function. Okay, so just to give a break to the speech what I'm giving, right? So we had seen that Sherrington identified a lot of differences between the functioning of a single neuron and the collection of neurons, which he called as reflex. And he explored the reflex properties by doing various experiments. No. So and now we will move on to the each of the properties slightly a bit in detail. So first property is that of latency, right? So I would like to ask you, right, especially the postgraduates, what is latency? If you're answering, it would be nice for us to interact.
Actually, it is the time period from when the stimulus is applied till the first initial response is seen. That is referred to as latency.
That's the period of latency. In the last class, Ramesh Babu said it explained about stimulus artifact. Right, whenever you give a stimulus, there will be a slight change in the resting state, and that's called as the artifact, stimulus artifact. From that to the starting of the stimulus is the latency. Right. So on the left-hand side, you can see the graph, right? We give a stimulus and there is a time gap, which is called as a latency. And what is the reason for the latency? The stimulus on the right-hand side, you can see that we have a stimulus. All right. And this stimulus, in order to cross the reflex pathway, it takes a period of time, and then finally it produces a response. So the time which is required between this is the latency. Okay. So what were the experiments that he did, right? One of the major experiment he did was he stimulated at different increasing intensities, and he tried identifying what happens to the latency. Right, in case of a nerve trunk conduction, the latency is very less, but here it is more. So he stimulated at different increasing intensity. What do you think we learned? What would happen to the latency? Yeah,
Hello.
can you please respond?
Hello.
Yeah.
So, sir, what I think is like if that is inversely proportional, like higher the intensity, the smaller the latency.
Latency. Okay, very good. All right. So the finding one was whenever he stimulated, right, the time period when he gave a small stimulus, and then when he gave a higher intensity of stimulus, and then when he gave a higher intensity of stimulus, he saw that the time period between the stimulus and the response was reducing. All right. So this is one thing which he found very unique to the reflex arc. Right? So he also did a second one. He also tried applying a stimulus when already a response was coming. And what he found was um, when already a response was coming, when he gave a second stimulus, again there was a time period, a small latency, after which the intensity of the response started increasing. This he called as a incremental stimulus. Okay, so he found that the property of latency is common to both the initial stimulus and also to the incremental stimulus. Okay, right. Okay, so before going to the next part, we saw that on the first part is latency, right? So higher the intensity of stimulus, shorter the latency, right? So now we can very well relate this particular property to a real life example. When you're having a dangerous stimuli, which is the intensity of the dangerous stimuli when it is less, we take more time. When the intensity of that stimulus is going to be very much higher, we need to respond very fast, right? So that we see also in our real life. The next is the property of after discharge, right? It is the opposite of latency. So can you tell me what is after discharge?
Sir, even after the stimulus is being removed, there is an amount of changes seen in the response in terms of amplitude.
Yeah, there is a response continues to come even after you stop the stimulus. That's your after discharge. Okay, right? So here I have compared two things. One on the left side is with the nerve trunk, and on the right side is the reflex. So when you give stimulus, right? When you give stimulus, right? You see that the stimulus time and the response time, right? Sorry, and the response time are same, right? So we have commonly seen that whenever we give an electrical stimulation to a patient, if you're going to keep electrical electrode, pen electrode to a nerve, and you will see that you get an immediate response in the muscle. Once you switch off or once you lift that pen electrode, you're not going to get a response, right? But when you see in a reflex, he saw that it was not the same. So he saw that when he gave a stimulus, right? Although the stimulus has stopped, as you can clearly see in this, right? As you can clearly see here, that's the stimulus duration. But you can see the response, the duration is very much higher, right? So this is the after discharge. So it is a stop time of the response, right? So when does it stop? We saw when does it start, and that was the latency. Now we are seeing when does it stop, and that is the after discharge. Okay. All right. Now, in case of a flexor, okay, right? When a noxious stimuli or a pinprick stimulus is applied, the limb is flexed, and immediately this flexion reflex, if it stops off, the limb will again come towards the noxious stimulus, but it is held in that position, right? So that's the importance of the after discharge, right? Now, what he says is when I stimulated a knee flexors with a Faraday current, once the current is stopped, even the tetanic response, which comes because of a Faraday current, stops within 150 time units. That's 150 sigma, right? But he says that in a flexor reflex, it often persists up to 5,000 sigma duration, right? So we clearly see here he has measured it, and he has told that um, the clear difference between that of a nerve conduction and that of a reflex, right? So in relation to after discharge, he conducted a few different experiments. Experiment number one was he was increasing the intensity of stimulus. Experiment number two was he was increasing the frequency of stimulus, right? And experiment number three was he studied it during flexion reflex, scratch reflex. So he studied it during different reflexes, okay, right? So what do you think would be the response when you increase the intensity of stimulus? What do you think will happen to the after discharge?
So it will increase.
The after discharge will increase. Any other change do you think might be happening? Okay. What do you think would happen when there is an increasing frequency of stimulus, when you're continuously giving it the stimulus? What do you think would happen to the after discharge?
So the response would sum together and increase in amplitude.
Amplitude? Okay, right. Okay. What do you think will be the difference between a flexion reflex and scratch reflex in terms of after discharge?
So in flexion reflex, it would be more than the scratch reflex.
Okay, right. So that's what we discussed, right? Mr. Subramanian, and we had a lot of discussion about that, right? And what is the purpose of that? So we'll look into the findings of it. Here, one is increasing intensity increases the duration of after discharge. Now, you can see the values which are given on the bottom of the slide, right? So he had increased the intensity of the stimulus, 69, 110, 190, 300. He has increased the intensity, and he measured the reflex, that is what is the intensity of the reflex which is happening, and also he analyzed what is the amount of after discharge which is happening, the duration of after discharge, right? In this picture, we can see very clearly here the stimulus has been given, and you can see that there are around 7 responses, 1, 2, 3, 4, 5, 6, and 7. And with each time when he has increased the intensity, you can see very clearly that the intensity of the response is increasing. But more strikingly, when you look at the after discharge, you can see that the stimulus 1 and 2, we see that the after discharge goes away immediately. But when you see as compared to 3, 4, 5, and 6, you see that the after discharge duration is all very much higher. That means that greater the intensity of stimulus, the longer the response of the reflex. In the meanwhile, you can also see that the after discharge is not very smooth. It is kinking up and down, right? And that was a clonus response, right? So that also happened. That's the other finding. Okay, right? The next finding is when you increase the frequency, right? The duration of the after discharge was increasing. That's what he found. Okay, right? Now here, the picture may not be very clear. I'll just know it, right? So we had the top one is very low frequency. You can see that there are two stimulus which are close by to each other. In the bottommost picture, you can see that the stimuli, one stimuli and the other stimuli, no, for so much time the stimuli had been um, conducted. So the first stimulus, first point of stimulus is the starting, and the second point is the ending of the stimulus. Okay, right? Clear? So when the frequency is there, in the first picture, you can see that the after discharge value is increasing slightly. Okay, right? You can see that on the topmost picture. One second. Yeah. On topmost picture, the after discharge is very much close by to the baseline, and as compared to this, you can see that it is higher. It means that it has been there for some time and it is continuing. So there is both an increase in the amplitude, the magnitude of the after discharge response, as well as the duration. Okay. So that's the finding 2, right? Finding 3 is he saw that with both flexion and scratch reflexes, they both demonstrate after discharge. That's the first finding, which means that after discharge is not to one reflex or the other reflex. It seems to be a common property of all the reflexes. And he saw that the duration of after discharge in a flexion reflex, right over here, is sustaining for a longer period of time, and the scratch reflex, it is ending it in a shorter period of time. And we had discussed what could be the plausible reasons for that. Okay, so that completes his lecture 1. It was about latency and after discharge. Now moving on to his lecture 2, right? The next property he tells us about summation. Right? Now, what is summation?
So it is the addition of the stimulus.
It's addition of the stimulus to what purpose?
In order to generate a greater response.
Generator?
Greater response.
Bigger response. Okay, right. So that's it, right. So how did he do the experiment? He provided repetitive stimuli to the reflex arc, right? And he studied when these repetitive stimuli are given, what happens to the output, right? So here I have compared 2 things. One is a simple nerve conduction. This is that of a simple nerve conduction, all right, and that across a reflex path. Okay, all right. So during a simple thing, right, here you can see that he had given lesser stimulus, a higher stimulus, and that's one aspect to it, right? And what we see is in a nerve trunk, all these belong to the nerve trunk, right? And each time he gives a stimulus, there is a response which is similar in feature. But when he repeats the stimulus, right, either of higher intensities or of similar intensities, he finds that the response to the first stimulus adds up with response to the second stimulus, and it becomes bigger. It adds up to the third stimulus and it becomes even bigger. So he said that when you are giving stimulus again and again, the subliminal stimulus, what happens is the responses are adding up together one after the other, and he called this as summation. Okay, right? And since he had to give it in quick succession, if he had given one stimulus and then if he had waited for a longer period of time and then he had given other stimulus, then it was not coming out very well. So it was not adding up. So he said it's called as a temporal summation. Quick succession in time, and you get a addition of responses. The other feature is also when you are giving a higher and higher intensity of stimulus, you in a nerve trunk, what happens is they follow all or none phenomena. So all the stimulus will produce a similar response, but a reflex arc acts differently. When you give a higher and higher stimulus, it produces higher and higher magnitude of responses, right? So that is also implicit from this. Okay. The next property he had explained is irreversibility of conduction. So on the left hand side, we have the nerve trunk where you give a stimulus and you see that the response goes both above towards the cell body, which is antidromic, and also towards the normal course of physiological conduction, which is called as orthodromic, right? But when you are going to give a stimulus in a reflex pathway, what happens is the law of forward direction, or the Bell-Megendie law, is followed, where it goes in only one direction, right? When Sherrington analyzed this, he thought that the reason for why it is going in one direction could be that the synaptic membrane might be more permeable in one direction, but it might not be, it might be very less permeable in the opposite direction. So because of it, it might not be going. We had also seen across the different studies that he compared it with two other functioning of two other systems. One is within the body, the other one is a sea animal, right? The medusa, or we call that as a jellyfish, right? So that, and the working of the heart. And he saw that in the medusa, it was when one particular direct when one particular stimulus is there, that is when it's trying to move ahead, the impulses were passing in all the directions, right? And that is why it could make the whole of the head of the jellyfish contract in one beat, right? But he found that when he compared that and he opened the nervous system of the Metsa, he found that all the nerves were continuous. But in our nervous system, in the reflex, there was a gap between one nerve and the other nerve. This also led to his increasing suspicion that the synapse might be a potential source where the property of the reflexes are being generated. Right. And at the end of it, he also saw the rhythm of response. What is a rhythm of response? Is if you're going to give a stimulus, say 1, 2, 3, 4, 5, 6, 7, then what should happen is each of these stimulus should produce a equal rhythm of response. So there should be obviously 8 responses. And this is what he saw in a nerve conduction. But when he had seen about the reflex pathway, he saw that the rhythm of stimulus was not matching with the rhythm of response. Whatever was the rate of stimulus, whether it was 5 times per second or 10 times or 100 times per second, he always saw that the rate of response was approximately 4 per second. Princey, I hope I'm right in my number.
Yes, it is.
Right. Okay, right. So what he said was, if you're giving a stimulus for very continuously, but if the response is not coming continuously, then there should be a place where it is breaking up. That means there should be a rest period. And that period he said as a refractory period. So he identified a phenomenon called as a refractory period during the giving continuous stimuli, right? And he said that he had done an experiment. We had seen his experiments where he gave a stimulus to the shoulder of a dog, and he was seeing the scratch reflex, and he was trying to measure the scratch reflex, and he found that the response was coming from the legs, and he found that it was connected through two major synapses, and along with the synapses there was a, gap or a rest period between one and the other stimulus which was going on. So he finally concluded that the synapse was playing a major role in this reflex functioning, and that was giving a gap. Even in a scratch reflex, when the person was trying to do very fast, we saw that there were a lot of gaps in between, which was there, right? Okay, so that completes the functioning of the simple reflex. And we saw that numerous differences were there between nerve conduction and reflex arc, which made him understand that reflex is some unique property, and that's the property of the nervous system. Okay, so I'll end my talk here. So we'll move on to just discussions. Yeah. Any interesting points for you to throw in? You can just throw in. So that's what we commonly teach as the reflex theory, right? So I told about it. Now the second part is the most interesting part, where he found that how the different reflexes are functioning, what are the outputs which are coming out of it. Yeah.
Hello, Vasant. I'm Ramesh talking.
Yeah, Ramesh.
Yeah, it's very well explained. Very nice. Good.
Yeah, thank
Just a few points like that.
Yeah.
Why that you told about the two-way connection, whether it is possible on either direction.
Okay.
Experimentally, when you stimulate a nerve fiber, if you stimulate the middle part of the nerve fiber, both side conduction is possible. But physiologically, functionally, in our body, usually one way it happens. So
Yeah, Ramesh, I'm not able to hear your voice.
according to my opinion, maybe because of the
Yeah, your voice is breaking
neurotransmitters.
out. Yeah, your voice is breaking. Yeah.
Yeah, network issue was there. So my another point is like uh, first I will explain a little bit about why there is a one-way direction at the synaptic level. The main reason is from presynaptic neuron to postsynaptic neuron. It cannot go back from postsynaptic to presynaptic. So that one-way connection is the release of the neurotransmitter and acting over the receptors and then producing
Yeah. Now,
excitatory or inhibitory potential.
yeah, Ramesh, one point of contention here.
Added up and
No, I'd like to tell one thing, right? We are discussing about a function which has happened, events which have happened 100 years back. And that's what Sherrington has understood at that particular point of time. And he explains that, right? So I think we are doing a time travel, right? And coming over to the current point of time and trying to understand it, where we have far more better understanding, right? So yeah, please
I think so.
continue with the explanation for that.
But then the delay, what I am expecting is, it's my opinion. Like, so when you give 3-4 stimulus, the presynaptic neuron, um, it has to act on the postsynaptic neuron or in the muscle, right? So initially, suppose one stimulus is given, sometimes we cannot say that all the stimulus will produce response immediately. So
Yeah.
the summation may be possible there. For overstimulation, it can produce an immediate summation. Then the muscle or the postsynaptic neuron will reach to the threshold level, and then it can produce a response. So that could be one of the reason why there is no equal response for the number of stimulus given.
Okay. I think we may need to again go back to Sherrington and read his experiment. Last time when we were discussing, we were seeing that whether these other reasons where how did he control for these things. So probably we may need to look into his original papers on whether he try to control it or not, or the responses reason might be this, and he might have wrongly concluded that it was the refractory period, right?
I'll just look at it.
Ramesh, I'm not getting you. Can you speak a bit louder?
I will have a look at and tell you. Okay.
Yeah. We'll together look into that aspect of it, right? Because yeah. Yeah. Ramesh, are you speaking? I'm not able to hear your voice.
No, it's fine.
Oh, okay. Fine. So the points
Mine
point.
over.
Yeah. Right. So
Yeah,
where
yeah,
where
there.
yeah, where we are going to go next is I would want Ramesh to please again continue with the beautiful lecture what you've started, and to understand the further processes which were there, and also I would like to understand about this current the method in which he has given, so we can slightly have more insight into his experiments on the make shock, break shock, and things like that, because he saw different response with the make shock and different response with break shock. So I'm really not very clear about it. So I want you to speak during the next session on that particular thing.
Yeah, sure. I will do that.
Yeah. Yeah. Others, yeah, I would like to ask opinion from our students first before going on to the other staff. Ria, Riddhi, uh, Suchi, are you there?
Yes, sir.
Hello.
Yeah. Yeah. So did you get a idea of what
Uh,
is the points which he had told?
Hello.
Yeah, Riddhi.
Yeah, sir. It was clear now, sir.
Okay, fine. Because the clarity is what we want you to build alongside reading the different experiments and understanding it.
Yeah, sir. Now it's bit clear.
Okay, fine. All right. So, anyone else? Sandal sir? Any points?
Yes, Sintan, it was clear now. We could understand, correlate all the lectures, what we came across so far, and collectively we could understand all the lectures now. And when we read motor control theories and all in the book, it is just few lines. It is explained. Now I wonder what amount of depth amount of work he has done, and it is explained
Yeah. Yeah,
in
that's
very detail. Okay,
that's why
all we
it has
can do
been
is
for a period
we
of
can
100
wonder.
years in physiotherapy.
Yeah, yeah.
Yeah,
100 years before, almost everything whatever we study in a modern physiology book already has explained in different way.
different way. Right?
Yeah.
He's done a very extensive work. Right? So I
Yes.
think there are two or three people who are like him. And if we read them, we will be able to better understand what we are doing at the current point of time and why we are
Okay.
doing it. Right. So that was very interesting. Right.
Yes.
Anyone else? Sarju, any points?
No, sir.
Okay, fine. Of course, many of you have detailed a lot of these points. You have explained it in great detail. What was this experiment? How did you do it? And what were the different findings? We had, in fact, listened to a lot of other things also before coming to the summary of it. So I'd like to thank everyone who actively participated, read 100 years back literature with so much of detail. I know that some of you have been cross-referencing things and you were not getting it, and then you were trying to understand it. It was a huge effort, and I think at the present point of time, our understanding about the simple reflex at least is simplistic. Okay. Yep. So yeah, if you don't have any final points, what I would like to tell is, yeah, Ria, anything? Nothing random. Okay. I
No,
would
sir.
ask, yeah, Raji, can you just share, send us in the group article which you had found, which was connecting between the older theories and this theory in detail.
Yeah, definitely. I'll send it in the group.
Okay, Right. So we're reading it, and she has found a very interesting piece of article, which had tried explaining what Sherrington had tried to do on. Okay, thank you all for joining. What I think of doing is there is still a few things. One is I think grading of responses. Mohammed Musin, you're going to do it, right? Yeah, Musin, I think.
Yes, sir.
Yeah, right. So grading of responses, that's one small part which is pending in simple reflexes. But then the next part is again coordination of simple reflex, but it's going to talk about more important reflexes where two or more sets of reflexes are going to combine with each other, and then they are going to work ahead. So we will continue that with compound reflexes. And I'd also like to take all of your opinion on uh, when shall we start with the compound reflexes, because we are now traveling time, we have time travel to 2020, and we are reading the current motor control literature. uh, what is your individual opinion? Should we also side by side continue to understand the compound reflex, because that is a very important for us to understand the current one? Or we can also, if you want, we can also concentrate on the motor control workshop which is going to come, and then we can also restart. However it is, I would be very comfortable with that. So also please do give your opinion when we are discussing in the group. Okay. Thank you all for joining, and I'll close the meeting for the day. Okay. Thank you.
Thank you very much, Sandan. Good
Thank you,
night.
sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.